Okay, good afternoon, everyone. Welcome to our webinar about bioaccumulation of marine and freshwater toxins. We'll just wait for a few minutes for everyone to join, and then we'll be able to start. Okay, um, so in this webinar, we dive deep into bioaccumulation of marine toxins and freshwater toxins, and we explore some of the causes of harmful algal blooms and what impacts these can have on human and animal lives. Please feel free to ask questions throughout the webinar using the Q&A function at the bottom of your screens. This webinar has been recorded, so you'll be able to access that later for future viewings. We're going to start today with a short poll, which you should see on your screens shortly. So the question is, do you have experience working with any of the presented toxins? Great, so we've got a really nice mix of answers here. Lots of paralytic shellfish toxins, some cyanotoxins. Brilliant. Just wait for those last few answers to come in. Great. So I'll just start with a few words about LGC. LGC was founded in 1842 as the laboratory of the Board of Excise for the UK to protect excise duty payable on tobacco importation. LGC was privatized in 1996, and today we provide customers with high quality reference materials from our comprehensive Dr. Ehrenstorfer range. We also offer portfolios from suppliers of high quality reference materials for specialist applications. In 2015, we became CIFCA's preferred partner, offering their marine and freshwater toxin reference materials to our customers. Today, our portfolio has expanded to adapt to changing regulations and technology, as we support your need for high-quality reference materials for food and environmental analysis. Currently, we offer more than 12,000 reference materials with many products available for testing of pesticides, pharmaceutical and veterinary compounds, and standards for regulatory methods. Over 45% of our products are accredited to ISO 17034 and produced in our centers of excellence located around the world. LGC selected CIFCA as a key supplier for marine toxins because CIFCA are specialists in the manufacture of marine toxin reference materials. They're one of the first companies in Spain to be accredited in ISO 17034, and the only one in production of marine biotoxin standards worldwide. CIFCA offers the highest possible confidence in its products thanks to its accreditation, which reflects the maximum competence both at a technical and managerial level. CIFCA's areas of expertise include agri-foods, textiles, cosmetics, and biopharmaceuticals, from the development of standards and reference materials to detection kits. CIFCA has been focused on the continuous process of improvement of procedures, working to achieve the highest degree of trust and reliability. They are committed to the quality of products and services. CIFCA's system of continuous improvement has led to excellence in services and processes to satisfy customer requirements and expectations, making them one of LGC's preferred partners. A bit of information about myself and our speaker today, Eva Seguide. I am the food product manager at LGC. I obtained my master's degree in chemistry in 2013. And from 2013 to 2019, I developed freeze-dried formulations for non-invasive prenatal diagnostics as part of an EU-funded research project. Eva obtained her BSc in biology in 2003 from the University of Santiago de Compostela. In 2004, she obtained a research fellowship at the Marine Toxins Group from the Department of Pharmacology of the University of Santiago de Compostela, Spain, and completed her PhD on the effect of emerging phycotoxins in human neuroblastoma cells in 2009. From 2008, she is employed as a researcher in Laboratories CIFCA, where she has been highly involved in the pharmacological applications of marine toxins, dinoflagellates, culture activities, purification processes, and R&D management. She coordinates the R&D activities at CIFCA. She has co-authored 19 articles, published in international scientific journals, and 4 book chapters about marine toxins. She has participated in more than 20 international congresses and has also been involved in more than 20 national and international research projects, managing 10 of them at CIFCA. Eva will start her presentation shortly, where she will introduce marine and freshwater toxins, discuss bioaccumulation of these toxins, as well as presenting some detailed case studies, after which point we will take your questions.
Thank you so much, Tom, for your nice presentation. So, good morning and good afternoon, everyone. Hope you can see my screen now. Thank you for joining today. We will start with a few concepts about bioaccumulation, which is a process of accumulation of chemicals in an organism that takes place if the rate of intake exceeds the rate of expression. Chemicals are introduced into the organism through exposure to the abiotic environment or as a dietary intake, also known as the trophic transfer. Whereas magnification is a process by which contaminant concentrations increase in the tissue of higher species of the food chain. As contaminated smaller organisms are consumed, chemicals are accumulated and magnified in the tissue and organs of larger animals. Animals at the top of the food chain, including humans, have a risk of accumulating significant concentrations of chemicals through biomagnified food chain. Since nutrition and food safety are 2 of the general concerns of human society, food quality has become a leading issue, either because of the increasing importance attached to local products or because of the presence of less organic contaminants such as pesticides, hormones, additives, or marine toxins, cyanotoxins, and microtoxins. We will focus on some of the main groups of toxin bioaccumulation process, which can be classified under environment: marine environments, freshwater environments, and also mycotoxins which contaminate the food through the food chain in process, transport, or storage. Starting on marine environments, within the 11 main groups of marine toxins, we can classify them into toxins that are regulated and those that are not regulated, namely the amnesic shellfish toxins causing amnesic shellfish poisoning, or ASP, also named domoic acid group, diuretic shellfish toxin causing diuretic shellfish poisoning, which includes ocadic acid and pectenotoxin groups, and also ciguatoxin lipophilic groups and aspirated shellfish toxins, which used to be included in the diuretic shellfish poisoning syndrome, but are no longer included in this group since they do not cause diarrhea. And finally, paralytic shellfish toxins causing paralytic shellfish poisonings. However, another marine toxins are focusing attention due to their emerging characteristics. Ciguatoxins, which cause Ciguatera fish poisoning, neurotoxic shellfish toxins, also known as the brevetoxin group, the polytoxin group, the cyclic amines groups, which includes pycnotoxins, spirolites, and gymnodimine, and finally the tetrodotoxin group. When establishing regulatory criteria and limits for marine toxins, various factors play a role, such as the availability of survey data, the availability of toxicological data, the distribution of the toxins throughout sample lots, and the stability in the samples, the availability of analytical methods and regulations already enforced in several countries. Starting on the first case, ciguatoxins, which cause the Ciguatera fish poisoning, while most of the marine toxin intoxications, such as PSPs, DSPs, ASP, NSP, or AZP, are caused by human consumption of contaminated shellfish products, Ciguatera is caused by the consumption of subtropical and tropical marine carnivorous fish that have accumulated Ciguatera toxins through the marine food chain. In last years, there has been an increase in global scientific interest of ciguatoxins related to bioaccumulation, triggering an analysis of its impact in a wide range of natural ecosystems. This can be related to the climate change and global warming, or the anthropogenic impacts. Ciguatera is caused by the consumption of subtropical and tropical marine carnivorous fish that have accumulated Ciguatera toxins through the marine food chain, and various aspects of these toxins will be reviewed in this presentation. Ciguatera is widespread in tropical and subtropical waters, being particularly common in the Pacific and Indian Oceans and the Caribbean Sea. The incidence and geographic distribution of Ciguatera poisoning are increasing, and newly recognized areas of risk include the Canary Islands, the Eastern Mediterranean, and the Western Gulf of Mexico. Medical practitioners must be aware that cases of Ciguatera fish poisoning acquired by travelers in endemic areas may present in non-endemic temperate areas. So species from the genera Gamberdiscus are the main responsible of these toxins. They may proliferate on dead coral reefs more effectively than other dinoflagellates, and the risk of Ciguatera poisoning is likely to increase as coral reefs deteriorate because of climate change, ocean acidification, offshore construction, and nutrient runoffs. These dinoflagellates are ingested by herbivorous or corallivorous species, and the toxins produced are modified and concentrated as they pass up the marine food chain to carnivorous species and finally to humans. Ciguatoxins and the closely related mycotoxin cause symptoms by interfering with ion channels on cell membranes, disrupting the signaling between nerves and muscles. The symptoms of Ciguatera typically last weeks or months, but in rare cases can become chronic. The European Food Safety Authority published in 2021 a report on the characterization of the risk of Ciguatera in Europe, confirming the emerging risk in the European Union. However, it is necessary to collect more data and standardized analysis methods or prediction models. Currently, a cell-based assay was established for evaluation of ciguatoxin-like toxicity in microalgae and fish, but it's important to highlight the lack of standards. Since during the transit through the food chain, an oxidative biotransformation of less oxidized toxins to the more oxidized and toxin ciguatoxins occurs. In the stomach of herbivorous species, partial transformation of Gamberdtoxins to ciguatoxins have been observed. The toxins, after their accumulation in herbivores, are transferred to other herbivorous species and are transferred to carnivorous species, which have been shown to have ciguatoxins but not Gamberdtoxins, indicating that any remaining Gamberdtoxin in the herbivorous prey is completely biotransformed in the carnivorous fish. It's noteworthy that ciguatoxin becomes more and more concentrated as it moves up the food chain, with levels up to 50 to 100 times more concentrated in the viscera, liver, and gonads. Experimental models show detectable ciguatoxins in muscle tissues after 2 weeks at levels above the threshold for human intoxication, 1.2 micrograms ciguatoxin 3 equivalents per kilogram. And results also imply that slow-growing fish may accumulate in their flesh higher ciguatoxin concentrations than fast-growing fishes, which has important implications for global seafood safety. The accumulation of toxin in fish becomes more pronounced with consecutive outbreaks, posing a health risk to fishermen, their families, and tourists. The density of Gambierdiscus populations within a given area and their toxin production could determine the eventual risk of Ciguatera in the area. However, the fact that the fishes are mobile, and the fact that the toxin in the fish could result from a long-term accumulation, their toxin content may not be representative of the concentration of microalgae at the time and place where the fish was caught. In example, several intoxications in North Florida have been related to migratory species from southern latitudes. The relationship between fish length, age, weight, and ciguatoxin concentration remains unconfirmed for the fish species. As a summary, there is a lack of official methods to manage Ciguatera. It is therefore not possible to test a representative sample of fish to determine the toxin potential of the remaining untested fish, even for a batch of fish caught on the same day from the same reef. The evaluation of the contents of ciguatoxins relies on the availability of standards and the development of sensitive and specific tools to detect them. There are 3 different groups of ciguatoxins that have been categorized based on their geographical location: Indian, Caribbean, and Pacific ciguatoxins. Each of these groups has a base chemical structure with minor variation in its structures, accounting for distinct differences in their toxicity. Today, more than 40 ciguatoxins have been identified, although less than half are structurally characterized due to insufficient amounts of purified toxin standards, which are in need for analysis. The production of 44-methylgambierone by more than 2,050 marine microalgal isolates consisting of 90 species from 32 genera across 7 classes was assessed by liquid chromatography tandem mass spectrometry. And it was discovered that the production of this secondary metabolite was ubiquitous to the 8 Gambierdiscus species tested. Following on from the recent structural elucidation of 44-methylgambierone is potential to contribute to Ciguatera intoxication events, and its applicability as biomarker compounds for Gambierdiscus was investigated. These results indicate that 44-methylgambierone cannot be used as a selective biomarker for toxic Gambierdiscus blooms. However, because of its considerable easier detection compared to ciguatoxin and microtoxins, it may still have use as a screening tool in high-risk areas to allow the early identification of benthic microalgal assemblages that could include toxic Gambierdiscus species. It is therefore advisable to invest management efforts both in preventive and palliative measures. The second case that we present is the paralytic shellfish poisoning caused by the saxitoxin group of toxins. It comprised a large number of analogs, more than 57, and produced by different dinoflagellates such as Alexandrium, Gymnodinium, or Goniaulax, causing paralysis by blocking sodium channels necessary for muscles to contract. The escalating incidence of paralytic shellfish poisoning is a global problem. Several decades ago, relatively few countries seem to be affected by these toxins, but in the last 45 years, most coastal countries are threatened by this group of toxins. the causes of this increase in harmful algal blooms is linked to global ocean warming and anthropogenic activity that promotes eutrophication, as well as the better managing procedures. This group of toxins comprises a high number of analogs with different toxicities due to the multiple bioconversion mechanisms among them. These bioconversions take place in seafood, but also on humans. After metabolization, especially on acid media, the toxicity is increased, resulting in a higher risk in humans. Therefore, it's necessary to control the presence of those analogs, which are regulated, involving the need of analytical standards or the improvement on new detection methods, such as UPLC with mass detection. This slide shows the biokinetics of mussels exposed to dinoflagellates under laboratory conditions for 8 days, then depurating them in clean seawater for 40 days. Distribution studies of the toxins in the mussels' organs reveal that mussels contain the majority of the toxins in the viscera. After a slight decrease in toxin contents observed during the first 2 days of the depuration period, the amount of PSTs present in the mussels dropped rapidly from day 2 to day 4. Later, they slowly decreased, being absent or detected in trace levels after 14 days in the depuration tanks. This is a high difference with Ciguatera, since accumulated ciguatoxins are slowly eliminated over time, with more yields estimated to depurate ciguatoxins with a half-life of 260 days. This information about predicted toxins levels in shellfish provides a scientific basis for assessing and managing PSP-associated risks. It should facilitate management decisions on closing and reopening shellfish beds during and after toxic algal blooms, especially in tropical regions for which relevant data are really scarce. Here we include also tetrodotoxin, one of the most potent neurotoxins, which was initially expected to occur only in pufferfish. A minimum lethal dose in humans was previously estimated at around 2 milligrams of tetrodotoxin. However, tetrodotoxin and analogs were recently detected in marine bivalves and gastrops on European waters, specifically in a gastrop from Portugal or in mussels and Pacific oysters from England. As a summary, despite there is an official method to manage PSTs, there is no currently regulation to manage tetrodotoxin. However, there have been several efforts to improve the methodology used to detect both groups of toxins. And a new PLC method with mass detection has been developed and tested on a collaborative study, which could improve the current methodologies. It is therefore relevant to take into account the potential of UPLC and SMS methods for the analysis of PSTs, analogs, and tetrodotoxin in shellfish tissues. Furthermore, the usual lack of standards is being surpassed in this case, being certified reference materials available now for analogs such as C3 and 4 and also tetrodotoxin, allowing the development of sensitive and specific tools to detect them. In freshwater environments, cyanobacteria, also called blue-green algae, can grow quickly or bloom when the water is warm, slow-moving, and full of nutrients. Cyanobacterial blooms are most commonly found in fresh water, such as lakes, rivers, and streams. Blooms can discolor the water and look like foam, scum, mud, or paint on the surface. These blooms sometimes produce toxins, namely cyanotoxins, that can cause illness. Structurally, the cyanotoxins are quite diverse, and then they can be categorized into 4 large groups according to their chemical structure. Cyclic peptides, such as microcystins and nodulains, which are both hepatotoxic cyclic peptides with 7 and 5 amino acids respectively. Alkaloids such as cyanotoxins, saxitoxins, cylindrospermopsin, limbiatoxins, and aplysiotoxins, lipopolysaccharides, and other bioactive compounds, such as neurotoxic amino acids, different from the 20 standard amino acids that comprise proteins, such as BMAA. Cyanobacteria are microscopic organisms that live primarily in fresh water and salt water at the surface and also below. As already indicated, they usually multiply and bloom under certain conditions. Here we can see a scheme of accumulation of microcystins in the aquatic food web, including main routes of biomagnification. The fate of microcystins at the end of the bloom event can include dilution with the water surrounding, deposition on sediments, biodegradation, or transfer into aquatic organisms. In general, the transfer of or accumulation of microcystins from one phase to other phases in the aquatic ecosystem is likely to be dependent on a number of factors, such as the microcystins concentration in the initial phase, the mode of transfer, and the biodilution capacity. These toxins are present in a global manner in all continents, being therefore a considerable risk. In spite of the high variability, zooplankton is clearly the best bioaccumulator of microcystins, reaching values over 1,000 micrograms per gram of dry weight. Biomagnification is the increase in microcystin concentration between two subsequent trophic levels, that is, between the prey and the consumer. Even if the high concentration of microcystins have been measured in aquatic consumers of different trophic levels, a meta-analysis based on the biomagnification factor quantitatively confirms biodilution of microcystins and not biomagnification as the dominant process in aquatic food webs. Kinetics of microcystin elimination after the transfer to clean water was studied in fish that naturally accumulated microcystins, specifically carps in a pond with microcystis species during 9 weeks accumulation period. Concentrations approximately an order of magnitude higher were determined in the hepatopancreas, which is the target organ for microcystins. The elimination experiments demonstrated that microcystin is rapidly removed from the tissues after the transfer of fish to clean water. It has been suggested that accumulated microcystin in edible fish may represent a risk to human health, and it has been demonstrated that microcystins are stable and not degraded by heat during cooking. In another study, lettuces were irrigated with contaminated water having 5 and 10 micrograms liter of microcystin for 7 days, which is the bioaccumulation phase, and lettuces rapidly accumulated microcystin per day after exposure to the toxins. Microcystin bioaccumulation in lettuce was dependent on exposure concentration and time, but became saturated after 30 days of uninterrupted exposure. After 7 days of irrigation with uncontaminated water, depuration of microcystin content in leaf tissues was 98 and 74% of the initial bioaccumulated values. The concentration of microcystin in lettuces as function of time indicates that at higher exposure concentration of microcystin, both and depuration are less efficient. According to a toxicological criterion, contamination is nearly eliminated after a time of 10 half-lives of uninterrupted contaminant-free uptake period, showing that it is possible to decontaminate microcystin from contaminated lettuce. So consumption of microcystin-contaminated lettuces constitutes a high-risk route for human intoxication. In fact, the daily consumption of 4 contaminated lettuce leaves could expose adults to significantly higher tolerable daily intake of microcystins than the ones set out by the WHO per body mass. Algae dietary supplements are marketed worldwide as natural health products. Although the properties have been claimed as beneficial to improve overall health, there has been several reports of contamination by cyanotoxins. These products generally contain non-toxic cyanobacteria, but the methods of cultivation in natural waters without appropriate quality controls allow the contamination by toxin producer species present in the natural environment. In the case of cylindrospermopsin, it is being recognized as another globally important risk of the freshwater algal toxins. Since 1999, a number of studies have demonstrated the ability for cylindrospermopsin to be accumulated in freshwater organisms. The global distribution of these toxins producer is growing, and despite the biomagnification factor is less than 1, it has important implications for human and ecological health risks. On the other side, no evidence for bioaccumulation of a toxin is available. So, as a summary, there is a lack of official methods to manage cyanotoxins, just being a guideline from the World Health Organization setting a limit of 1 microgram liter of microcystin. However, the evaluation of cyanotoxins content can be carried out with several methods, from screening methods based on functionality to confirmatory methods based on analytical techniques. The evaluation of the contents of cyanotoxins relies on the availability of standards and the development of sensitive and specific tools to detect them. There are several groups of cyanotoxins that has been categorized based on their chemical structure, and therefore specific methods for each case must be implemented. It's advisable to invest management efforts not only in the producer organisms, but also on the toxin production, since the toxicity may vary, and not all the species produce toxins during the blooms. So it's important to take in account the implementation of analytical methods, both in preventive and palliative measures. Furthermore, a harmonization among countries should be established, especially considering the current availability of standards.
Many thanks, Ava, for an excellent presentation. To summarize, marine toxins accumulate in the food chain from fish and bivalves through to humans, and here's a summary of the regulation. Contamination can also occur through consumption of vegetables irrigated with contaminated water. Global ocean warming promotes occurrences of paralytic shellfish poisoning as temperature climates expand. Many toxins do not have current regulations despite a need to monitor and test for these emerging toxins. And more and better reference materials will enable appropriate determination of the toxins in marine and freshwater. Before we move to some of the questions that you've been asking in the Q&A section, we have one final poll question for you. Is it important for you to use standards accredited to ISO 17034? So if you could answer this poll question, then we'll move on to the Q&A section. We'll just wait for those last few answers to come in. Great, thank you. So looking to the Q&A box, I think the first one, Ava, in your experience, do you think there is any risk on those cases where there is no legislation?
Of course, always exists some kind of risk, especially in the case of poorly studied toxins or in the case of tetrodotoxin, where the consequences may be very drastic. However, as indicated during the presentation, we are trying to contribute to the scientific community by developing standards for those toxins, and this could help to develop and harmonize methods of detection to be implemented.
That's great. Thank you, Eva. And what are the side effects or risks of things like microalgae superfoods?
Well, with superfood, I presume you mean the dietary supplements that we talk about in the presentation. It is important to know the source of use of the algae diet, since most of the toxic algae become toxic when they reach the bloom conditions in aquatic system. So National Institutes of Health warns that if you consume chlorella or spirulina harvested from polluted sources, you may consume toxic heavy metals. However, also cyanotoxins can be consumed, and all those risks should be controlled in these new foods.
That's great. Thank you. Could you share a bit more information about how you're characterizing the standards according to ISO 17034?
Well, the standards are characterized by two methods, as indicated in the ISO. One of them is the NMR characterization, and the other method is HPLC, usually with mass detection. And a combination of both techniques is used to indicate the purity, and just the information about the NMR is considered for the quantification of the value, of the certified value, which is its concentration. And those standards along the life of the batch, they are also characterized, well, firstly with the homogeneity studies, and along the life of the batch with the stability studies to confirm their stability and that there are no changes in the certified value. When there is some kind of change, because the uncertainty from the long-term studies is considered, a new revision of the certificate is released, taking into account that factor.
That's great. Thank you, Eva. So the next question is, are there any plans to expand the spectrum of cyanotoxins? For example, offering more microcystins, maybe desmethylated ones, other anotoxins such as dihydroanotoxin A, homoanotoxin A, or ling biotoxins, or
Yes, we first started with marine toxins. In fact, our accreditation scope covered initially the marine toxin area. Now it is expanded also to the cyanotoxins, and that was also a great effort to take in account. And yes, we are working with new cyanotoxins project. For most of those mentioned are in progress, but the stability issues must be taken in account because of the ISO. And there are some kind of tricky issues on the solvents and the long-term stability. So they are not being released in a short term, but we are working on them. Maybe in a medium term, they could be released.
That's great, thank you. Which regulations are more important for the end users? Are they applicable globally or just for individual countries? I think the EU has some kind of blanket for their own region.
Well, I think that could be discussed between, well, among all the community that we are here, no? Of course, for the end user, the best regulation to take in account is the one in force in its own country, because probably that regulation will take in account the habits and the most potential toxins that could appear in their area. But of course, a harmonization should be considered, as indicated on the presentation, because of course there are traveling issues, and the globaling the globaling effect is something to take in account, and the harmonization between among regulations in different countries should be considered also. Sure.
Great, thank you. There's one question which is a follow-up to something that you mentioned before. Since the concentrations are very low, could you share a bit more information about how you're performing the NMR testing?
The first batch, I mean,
do you have specific methods that you're following, or are they things that you've developed in-house at Civca?
Yes, of course, the NMR method that you follow is a validated method that we did in-house. Maybe since it's a very technical question, it would be better to answer by email.
Yeah.
But you have the first, the quantities that you will use for your batch. The whole quantity is the one that is quantified and characterized with the NMR. But I will try to get a further explanation or more technical explanation on this issue by email if it's fine for you.
That's great. Thank you, Ava. Yes, with all of the questions, we will provide a Q&A sheet at the end with all of the questions and answers, and any that we've not been able to get to. So you'll have a copy of the webinar as well, and all of the questions and answers that have been provided during this talk. I think that's all of the questions that we have so far in the chat. So I just have a few other pieces that we can talk about. So on the LGC website, we have a selection of webinars, white papers, and articles available on demand for food and environmental applications. We've got a variety of articles discussing bioaccumulation, dyes, and pesticide testing, and they can all be accessed here. And we also have a supporting product range for marine toxins, including cyanotoxins, calibration solutions, and some matrix materials. And you can learn more about how LGC and Civca can support your analytical testing by downloading this new brochure. That brings the questions section to a close. If you do have questions after this session, please feel free to contact us via our social media accounts. You can reach us through LinkedIn, Twitter here. And or you can ask us questions by, directly. I've also included Ava's LinkedIn page and also my own as well. So, thank you so much, Ava, for a really wonderful presentation and really great answers to the questions as well. It's been really brilliant. Thank you.
Thank you also. And thank you all the attendees for spending some time with us.
Great, thank you so much.